Okay, here we have figure 2 of the Stoeger et al. Paper, where they wanted to see if the general trend that they were finding of this like inverse correlation between transcript length and expression with age could be observed in other projects and other experiments. So I'm going to start with the first panel, panel A. Here they found basically, let me move my face down here. Where we go. Here they found 4 other papers that ran a similar experiment doing bulk RNA sequencing for different various tissues for either matched varying age mice or rats in the case of this Shavlakadze, I can't pronounce this name, et al., or the Reichwald et al. With killifish. So here we have the heat plot of those Spearman correlations, which I explained earlier, where they basically generated a correlation between transcript length and the relative fold change of sort of a pairwise comparison of different aged mice. If these cells are in yellow, it means that that particular tissue wasn't studied in that experiment. If it's gray, that means that the rho value was not statistically significant. If it's red, it means that there's actually an increased expression relationship between transcript length and age. So the older the mouse had generally higher expression of longer transcripts. And then in blue, we have that sort of anti-correlation that they generally found in their different tissues. So in the y-axis of this plot, we have all of the different tissues tested in these various experiments. And in the x-axis, we have this current study that I'm describing here, and then the 4 other projects. So what we see from this heat plot is generally for the tissues that were studied, we're seeing this trend of most tissues having this profile of decreased expression of longer transcripts, with a few exceptions in like the white blood cells and the liver and the kidney and skin and brain also, which is interesting. They basically summarized those findings in this bar plot. So we have the different correlations sort of broken down by not significant in the middle, long transcripts having a relative fold increase with age, and then the long transcripts with a relative fold decrease in age. In black, we have the different tissues studied in this project, and in orange, we have all the other studies that they looked at. So we're seeing generally that there's this profile of decreased expression in most tissues observed in the papers that they were able to sample from the literature. Next, they wanted to see if that profile was sort of driven in each tissue by a particular cell type. So they found a couple single cell sequencing experiments. And here we have a very similar bar plot for 3 different projects using various different single cell sequencing approaches that I'm not going to get into. But in general, they're finding that similar profile where most cell types have this sort of decreased expression profile, suggesting that these tissues have more of sort of a global reduction in long transcript expression with age, rather than one particular cell type driving it in different tissues. And then lastly, they looked at this tabula muris senis, which is this project that is trying to sort of compile all of the aging data sets that have been generated with mice. And they wanted to see if there are particular tissues that have a like profile of expression difference, or like a correlation profile that is like much more significant than the other tissues. So here we have sort of rank order of the sort of median Spearman row for each of the tissues. And they found they have sort of a different count of pairwise comparisons for the different tissues from this tabula muris senis project. And we're seeing that all of the tissues in this plot, at least except for the brain non-myeloid, which I believe are neurons, have this profile of reduced expression of longer transcripts, but they all seem to fall within a distribution that is comparable. So there weren't any really exceptionally like decreased expression profiles of these tissues, suggesting that they all sort of fall within a similar range.